Hello, my name is Philip Campo. I'm the Assistant Department Head for Education in the Computational Biology Department at Carnegie Mellon University. And that's certainly a mouthful, but what it means is in part that I work as the Program Director for our Computational Biology major. And so I'd like to tell you a little bit about that major today and a lot of the fun things that we do with students and kind of a big picture of what computational biology is. And I choose the penguin metaphor here because we're really the first computational biology major that you can do in a computer science school. And for that matter, we were the second major in the School of Computer Science at Carnegie Mellon. You could only major in computer science for about 30 years here if you wanted to study computation. And starting a few years ago, we realized that we wanted to broaden what we offer in the School of Computer Science. And so now we have a major in computational biology. We were the second major that you could complete. And hopefully over the coming years, you'll start to see a few more majors now that we have artificial intelligence and human-computer interaction as well. So I borrowed the penguin analogy from Randy Posch, who was an instructor at Carnegie Mellon and sadly passed away too young. But he had a great analogy of in a group of penguins, any penguin, one penguin has to be the first to jump off the iceberg into the water. So he used this analogy to inspire his students. And it's an image you can still see on the CMU campus today. There's a bridge from the School of Computer Science to the drama department that has a bunch of panels, and each panel has a single penguin on it in his honor. So we're, in a sense, the penguin to jump off and try the waters to broaden what we do in the School of Computer Science, as well as to offer a computational biology major to computationally minded students. And we're still looking for the other penguins to join us and jump off as we continue to recruit students. And so I want to talk about three things today. I want to talk about first, what the heck is computational biology? Since if you're watching this, there's a good chance that you know a little about computational biology, and I think there's probably a good chance that this might be your first introduction to computational biology. So if that's the case, I want to spend a little bit of time talking about what are the big picture problems that we study in computational biology as of today. So here is a classic problem in the field where we want to say, what is it that your DNA says about you? Or for a species, what is it that corresponds to the DNA of a species? So for example, you can consider lots of medical problems that have to bear on this, and I'll discuss that very briefly in a minute. But most people don't realize that this is ultimately a computational problem. So if, for example, we want to know what is the DNA in your genome in each one of your cells, what are the 3 billion letters of A's, C's, G's, and T's that make you a unique organism on this planet? Well, how we can do that is we can take a sample of your cells, we can apply some technique that will break your DNA up into small fragments, so they may be a few hundred or a few thousand nucleotides long. And then there has been an enormous amount of investment in lab techniques that will allow us to read these short fragments. So we still can't read a very long fragment of DNA on the order of millions of nucleotides, but we can read shorter fragments for very technical reasons. And so once we have those letters, the A's, C's, G's, and T's, once we know those sequencing reads, we want to then use the overlap. Because we had multiple original copies of our genome, we want to assemble the genome using those overlapping fragments. So here you can see that the orange fragment overlaps heavily with the blue fragment, and it overlaps heavily with the green fragment. And now at the bottom we can start assembling that genome shown in black. The critical part is it shows that just in one slide you see the balance between laboratory techniques and computational approaches. This actually cost $3 billion just for the first human genome, and many more billions of dollars have been spent over the past 2 decades in terms of making this process a lot more high fidelity, i.e. much more accurate, faster, and cheaper as well. So we're now at the point where we can sequence a human genome for on the order of about $1,000, where it used to cost $3 billion. So it's this remarkable moonshot human achievement. And it boils down to being able to take those overlapping fragments of DNA and assemble our genome using that overlapping information. So that's a computational problem. And if you think it's an easy problem, then you're unfortunately mistaken, because you have to handle a lot of noise in the data. You have an enormous data set. You're going to have potentially hundreds of millions or over 1 billion of those sequencing reads for certain organisms. And you have to be able to manage this data and figure out how to put this puzzle back together in a natural way, very efficiently with a computer. So that is a classic problem in computational biology. And I can kind of tweak this very slightly and show you a more modern problem in computational biology, which is, say you do what we have done with our students, whether it's our high school program that we run every summer for our students, or whether it's students in our laboratory course that is based off of quantitative methods in lab biology, where we go out to Pittsburgh's Three Rivers and we sample river water from a lot of different locations in Pittsburgh's Three Rivers over different seasons. And in that case, when we scoop out the water, the DNA that's present in that sample is going to correspond to a lot of microscopic organisms, especially a lot of bacteria. So the word identical goes away if I then apply this same technique. What happens if I shatter the genome into reads? Now it's genomes. And what happens then when I sequence those reads? What are the types of analyses that I then carry out? How can I figure out what all the organisms are that are present in that sample? How do I differentiate that from one sample to another? Maybe they're at different points in a river. Maybe they're at different points, different time points throughout the seasons. And this problem is far more general than just river water. You can, for example, see this same problem in many, many different clinical studies that are looking at what are the microbes that are present in your body. So this is another biological fact you might not know. Probably over half of the cells in your body are not yours, they're bacterial. And there are many research projects going on that are trying to sample these from different locations in the body, figure out how the bacteria are different from one person to the next, and then try and figure out how this might be implicated in human diseases as well. So it's a fascinating research problem that's still very modern. And of course, a lot of computational people will look at biology and say, well, once we can figure out the blueprint or the DNA of a living thing, we've solved biology. It's the source code of our bodies. We can sequence the DNA. We just have to figure out what the language of DNA is. And the biologist then pushes back a little bit and says, well, there's a ton of feedback and external processing. And if you want a good comparison, look at the source code of google.com. And you ultimately see just a few years of optimization by the developers at Google. Well, DNA is a process of optimization that's been going on for millions and millions and millions of years, 4 billion years, in every living thing, running in parallel. All right, and so now the computational person says, wow, biology is impossible. I wouldn't say that it's impossible, but it's a very exciting, very modern discipline that is waiting for computation in many cases to come and continue to revolutionize it, because the revolution is very much ongoing. You might say, well, why do we sequence genomes? Well, here's a great example. I mentioned bacteria. You might say, I don't think bacteria are that cool. But here's Darwin's notebook in 1837. It's the first example of anyone drawing an evolutionary tree, because he reckoned that if the species are differentiating based off of natural selection, it goes to reason that they would branch out over time as they differentiate. And so the natural construct there is an evolutionary tree. If you look at the plot on the right, I call that the second most important evolutionary tree ever drawn after Darwin. It's from a 2016 research paper where they took the same gene in many species, and this is a simplified version of the evolutionary tree, but you take the same gene in every species, produce an evolutionary tree for it, and what you see is that tiny green sliver down there in the bottom right is eukaryotes. That's everything you have seen that's alive on this planet. So most of what we think of as life, when you watch a nature show and everything there is a eukaryote. So you go outside in your garden, it's a eukaryote. All of us were eukaryotes. Archaea make up a larger piece of that tree, but by far the biggest, most diverse, most interesting piece is bacteria. And that kind of makes sense, really, because they can live in a lot of environments that we cannot. They can produce a lot of things like antibiotics that we don't produce, that we have to borrow from them. And so you get these type of revolutionary rethinkings of biology if you're able to move towards a data-driven science, and that's what biology is today. And then you might ask, how did they produce this evolutionary tree? And you could consult literally a thick textbook of different algorithms that may need to be applied in different circumstances, and that you really need to understand that rely on very advanced mathematics in some cases, or statistics, in order to produce a reliable evolutionary tree. So there's computational biology again. You might say, well, why do I care in terms of on a human level? This is actually the first person whose life was ever saved as a result of genome sequencing, a boy who had had, I think, dozens of surgeries. Doctors couldn't figure out what was wrong with him. As a result, they sequenced his genome, found that he was defective for a given gene, and then gave him essentially gene therapy for the gene that he was lacking, and it made him better. So they found what was wrong with his genome, that he had a genetic disorder that was very rare, and they were able to give him therapy for exactly what he needed. The hope is that this type of thing will be much more commonplace in 21st century medicine, that you might be able to use this blueprint, if we can figure out what the source code really means, as a weapon against disease, that you would have a map of what your future medical history might be. And so you could use that with your doctor to start trying and heading off diseases that you wouldn't know might be around the corner. There's also exciting gene editing technologies that rely on a lot of understanding all this and computational approaches. We're now at the point where, because there's a Dwayne Johnson movie about gene editing, I can assume that many people have become familiar a little bit with gene editing. But beyond the headline grabbing things like designer babies, you also have very practical reasons for why you would want to edit genes. One would be, well, imagine if we can start editing the genes of crops to be higher yield, tastier, healthier, and to be able to get those crops into a supermarket around the world for a low cost. We have 10,000 people in the US who die every year waiting on an organ transplant in the hospital. Imagine what we could do if, for example, you could unhook a pig heart and hook it up into a human. It might sound crazy, but that is functionally possible. So we slaughter millions of pigs, for example, every year in the US alone. And the heart will function if you transplant a pig heart into a human. But the problem is that there are remnants of viruses that have attacked the pigs over time, and the human immune system will recognize those parts of the DNA as invaders, and so you have kind of an immune system response against the heart. Now, what would happen though if you just edited out all those problematic parts of the pig genome and made it look more human to the human immune system? That's a very active area of research. It sounds like science fiction, but a lot of people are hopeful that it's actually going to be something that we roll out with the goal that at some point in the 21st century, no one needs to die in a hospital waiting on an organ transplant because they don't have a donor. One more would be that we can move beyond the DNA level. All right, and so computational biology has now grown far beyond just the study of DNA. The next thing that we might look at is being able to understand, well, how is it that the genes themselves are turned on and off at different levels, at different time points, with respect to different stimuli? So for example, all of your cells have the same DNA, but the genes themselves get turned on and off like this big soundboard at different levels, depending on whether, for example, it's a bladder cell or a T cell or an epidermal cell or an endothelial cell, etc. Okay, so you have all these different types of cells. And how is it that your liver cells and your bladder cells and your heart cells are different in terms of how they turn these genes on and off? Well, some statistical and machine learning approaches have only really recently, along with research technologies that will allow us to measure the soundboard levels in a single cell. These are allowing us to produce beautiful charts like what you see here on the left. This is from the Chan Zuckerberg Biohub, so that comes from Facebook funding via Zuckerberg, where they were able to, again, using stats and machine learning, differentiate different cells in the mouse and see clustering based off of understanding how these levels of gene expression are turned up and down in different cells. That's very powerful, and of course it required a lot of biological knowledge too. But it's a very powerful thing to do because now you can say, well, how does those plots look like in a diseased mouse? How is it that they compare to humans? What about a healthy human versus a diseased human? Can we understand that much better? Because we're still at the beginning of getting this framework. At CMU, we have relatively recently gotten a piece of a $55 million or $54 million NIH grant where we're working on the software used for kind of producing software for helping biologists in the lab understand, after they've produced a data set, be able to work with that data set and understand the analysis computationally of it. Drug discovery is another area that needs computation very badly. So in computer science, you may have heard of Moore's law. In drug discovery, over the past several decades, we've had Irum's law, which is Moore's law backwards. And so it says that per dollar spent, you are getting an exponentially decaying number of drugs. So in terms of inflation-adjusted dollars, we used to spend about $1 billion and we would get about 100 drugs out. That was in the 50s. So if you imagine this pipeline is putting money in and getting drugs out on the other end after clinical trials and regulation. Now we spend $1 billion and we get on average one or fewer drug. So we get 1% for the same investment in research and development in terms of drugs falling out the other end of the pipeline. There's hope that this is getting a little bit better with computational approaches, say for example simulation. I'll show an animation of ultimately what a lot of understanding how drugs, say, interact with potential protein targets boils down to. So here's a classic problem in computational biology where I may have a protein that is represented as a strand of amino acids from a 20-character alphabet. And then but via some process, this strand forms into a three-dimensional protein shape. Even if you untangle the protein, right, or just sequence the strand, it will essentially always form into that three-dimensional shape. So there's a clear problem here, which is to understand how the order of amino acids somehow determines this final three-dimensional shape. If we can understand that, we have a lot more understanding of how things interact on the molecular level. We can start building better and better simulations, say for drug discovery, and this is a problem that a lot of people are working on today. It's also a problem that is not new. The Russian Academy of Sciences Protein Institute has been working on this for over 50 years. So it was founded in 1967, and they call it fundamental research on the protein problem. And so that means figuring out how a protein is forming into a three-dimensional shape and how it's interacting with its environment. There's another area of computational biology, which is analyzing medical images. We're now at about 20,000 petabytes of medical data worldwide, and most of it, about 90%, is stored in images. So here's a computational problem for you. If I give you a lot of these images, say they're images of skin cells with lesions on them. Let's see if you can train a computer to beat a doctor's diagnosis of those skin cells. So there are two examples of where this has been applied in a really cool fashion. One is with skin cancer, and the other is with a condition called diabetic retinopathy, where algorithms have now been developed that can look at these images and diagnose the condition of, say, a cancerous skin lesion or diabetic retinopathy better than a doctor. And so we are hopeful that every condition may start to see automated diagnoses that will help us reduce medical costs and improve patient health. There is one caveat I should say there, which is that if you ask one of the best dermatologists in the US to classify a skin lesion, for example, as benign, cancerous, or growing unexpectedly but not necessarily cancerous, so you have three classes that you've got to put all your images into, a doctor gets that right about 2 times out of 3. So this is part of why you have so many biopsies, because the human eye can be tricked by skin cancer. The algorithm, you say, how accurate is it? Well, the state-of-the-art algorithm, I think, is currently about 70% accurate. So it does beat a doctor currently, but it's not beating a doctor to such an extent that we're going to just turn everything over to a computer. We still have so much work to do in understanding the underlying biology and medicine and applying this to real world problems with computation. Cellular images are another area where you might think everything has been understood, but we are still seeing revolutionary developments in microscopy. Here's, for example, something that two of our alumni working at the Allen Cell Institute in Seattle produced. They were looking at bright field, very simplistic images of cells. What's nice about these is even though they're simplistic, and you're just essentially shining a light at the cells and getting this black and white picture, you're allowed to you don't have to kill the cells. So you're able to see the cells in their living environment. Now, what are these colors? These colors are a deep learning model that they trained that was able to infer different levels of organization, whether it's seeing what the DNA is or seeing where the cell membrane is in these very simple images. And they're even able to use this so they can see this at different layers of the cell. And so this is amazing because it's more really than what the human eye can see. It's bringing this out and even inferring the 3D shape of these cells to let researchers see what they couldn't see before with this type of microscopy. So it's an amazing development to be able to see all that hierarchy from such simple images. And it's cool because our alums did that. The next thing I would show you would be, well, here's a hot take that somebody fortunately had in 2017 in a short paper that they published that I don't have to propose. I can simply point to somebody else where they say, well, biology used to be this very fixed discipline and everything was experimental and everything had to be done in the lab, and there was this random little offshoot called computational biology, but that is no longer the case today. Biology is now fundamentally a data science, and if you're working in the lab, you're probably generating these very large data sets, whether it's images or DNA or protein analysis. And it would be very strange for you to not be well versed in computational approaches for analyzing it, as well as understanding that we still have very many unsolved problems in biology, and it's hard to see how we're going to solve them without computation. So it's an unbelievably exciting field to be in. Now, that's part one of my talk, and I love telling students about kind of what computational biology is, because what biology is thought of and what the frontier of biological research actually are. There's such a big difference there, and it's so cool getting to connect students to that. In part two, I would tell you a little bit about our computational biology major and ways of studying computational biology at CMU. So the first picture I would show, we've got these honeycombs. So all of our majors in the School of Computer Science have these honeycombs. We started these honeycombs with the thinking that, well, computational biology is sitting at the intersection of a lot of different things. So we should have a core of coursework, and it should pull from different cores of study. And some of these cores of study are going to be analogous to other courses that other students at the School of Computer Science take. So for example, we need to have a CS core in our major, and that's very similar to the CS core coursework that a CS major or an AI major or a human computer interaction or whatever SCS major would take. We also have analogous mathematics and statistics. And of course we have the same humanities requirements too. So that's that orange hexagon is the general education where everyone has certain humanities and arts requirements as part of their degree to round out their degree as well. So those three cores are going to be very similar among the majors, if not identical. Our core is going to pull coursework from those areas. So if you take a course like computational genomics, it requires our students to have taken several mathematics courses, including probability and statistics. Students will have typically taken 1 or 2 biology courses at that point as well. And students will be strong programmers and will have taken all the way up to Introduction to Machine Learning. And so that course now is able to assume that wealth of background from students. And so it's taught at a very high level. It's a course that, with this depth of understanding of statistics and machine learning and computation, you essentially will not find at an undergrad level at any other university. And so we're very proud of being able to rely on how very strong our students are mathematically and computationally, and leveraging the fact that they also take a few biology courses to understand the fundamentals of biology that are going into the problems that we solve. The next question I get from a lot of people is, could I major in another SCS major and computational biology? And so the answer there is yes. So students feel like they want to try every possibility on the menu of everything that we offer, and that's great. And so we try to find ways to facilitate that. If you're outside of the School of Computer Science, it's possible to transfer in. I'll say a little more about doing a double major inside of SCS momentarily, but we require students who are coming from outside SCS to have an overall, we call it QPA, you can think of it as GPA, QPA of 3.0 or better, and then we have 6 gauntlet courses of a QPA of at least 3.6. So here we have a mathematics course that's taught in the CS department, 3 computer science courses, a biology course, and then our introductory computational biology course. And so essentially students need 4 A's and 2 B's or equivalent in those courses, and these are all very challenging courses. And so admission from outside of the School of Computer Science isn't guaranteed. This transfer is a very high bar, unfortunately, because we have high demand for all of our SCS majors. We do have an additional major though, and this is open to all CMU students. It's also a good fit for other majors in SCS. And so if you're inside of CMU and you're an AI major, for example, and you also want to do an additional major in computational biology, it's just a matter of clicking a button and you can sign up for that. If you're outside the college, say you're a biology major or you're in the engineering school, or heck, you're a music or a drama major, you're also welcome to do this program. You just need a 3.0 QPA and a few prerequisite courses to do that. So you would keep your original major and then you would take a few additional courses or several additional courses in order to complete our additional major in computational biology. We also have kind of lower requirements in terms of not having to do an entire major. So if you're outside of the School of Computer Science, we have a minor available for you. And if you're inside the School of Computer Science, we have a concentration available. So that's tacked on to your degree. So it's not as much as an additional major, but your degree would show that you have a concentration in computational biology. And that's one of the family of concentrations that we offer that are open to all of our SCS students. Both the minor and the concentration are 5 courses of coursework. And then the next question that people ask is, well, in terms of the nuts and bolts, how is it that if I came in and I wanted to see if I liked computational biology, try it out, or maybe I know I don't want to be a major or a concentration or a minor, but I at least want to take a course, what could I do? And so School of Computer Science students, we suggest to take the same first semester of coursework. Everybody has the same wonderful first year advisor, and everybody is viewed as a School of Computer Science student first. And so you'll have the same fall coursework. You may place out of a course or 2 here or there, and then there will be replacement courses. So if you place out of a calculus course, then you can take a replacement, more advanced mathematics course, for example. You may plug in a humanities course or a science course there in your fall. We have very tailored advising, but this is kind of the standard series of sequence of courses we would suggest for students in the fall. And then we ask all of our students who are interested in Comp Bio, well, take our Great Ideas in Comp Bio course. It's a second semester, i.e. first year spring course. You won't find a course like this anywhere else, where we get students doing computational biology as a first year undergraduate. That doesn't exist at other universities. And it explores what are the great ideas that have made us into our own discipline. What has revolutionized biology computationally? And the things that I talked about before, evolutionary tree construction, assembling genomes, metagenomics, where we try and figure out what are the species present in a sample that may have thousands of species, and differentiating RNA or gene soundboard levels between 2 individuals. Those are the types of problems and more we talk about in that class, and we connect students to the kind of the big work that has made us our own field so that they can get a flavor of that in their first year. We also suggest that students take our excellent modern biology course at some point in their first year. It doesn't have to be before our Comp Bio course, and so we would suggest it as a spring course at the latest. And then all of our students in the School of Computer Science will declare a major in March around the time of spring break. And so regardless of what you're thinking when you enter, everybody has a chance to declare whatever major they like in March around spring break of the first year. So that's earlier than at some universities where major declarations may happen in, say, a sophomore year, But at CMU, where we're on the college model, we know our students are interested in something computational, and so we ask students to declare their major in the spring of their first year. There is some fluidity to that. So for example, we've had students go from CB to CS and vice versa, and it's not a complicated process either, so you're not locking yourself into an eternal decision by majoring in one major or another. But typically students get a good sense of what they want to do in their first year and stick to it, so we're happy about that. If you happen to be in the process of applying currently or interested in applying to the School of Computer Science and Computational Biology, we have a checkbox that you can select on the Common Application when you're doing that application process. And you might wonder, well, what are we looking for in terms of our students? Students who are great fits for us, you might think, do I have to be an expert coder? And you don't have to be an expert coder to be successful in any of our majors in the School of Computer Science. And we would back that up as well for computational biology. So if you have some computational skills, you've been coding since you were 3 or 4 years old, fine. I'm sure that's a plus. But what we're looking for students are students who are very strong in math quantitatively, and students who love biology. So if you love biology and you're strong at math and you want to see a very modern presentation of life sciences as a data science, then we think you're a great fit for us, even if you haven't programmed before. Right? You can take an intro programming course your first semester here, and about 1/3 of our students in the School of Computer Science do, and they all do great. What makes part 3 of this is, so I've talked to you about what the heck is Comp Bio. I've talked to you a little about, what are the logistical nuts and bolts of our degree. And part 3 is what makes us a unique major outside of the classroom. And the first thing I would tell you is there's me in a pink shirt instead of a blue shirt. But we like to have fun with our students. We have ice cream socials with our students and lunches and cookouts. We take our students to Kennywood Amusement Park for Fright Nights in October, where there's a Halloween theme, so you get roller coasters open, but you also get scary haunted houses and so on. We do this in all seasons, so we do in the winter time, even when it's cold, we'll go out and take advantage of, make the most of Pittsburgh winters and go to a local park and do snow tubing with our students. And so we love having fun with our students, first and foremost. We like to have a relationship with them that's more than just a classroom relationship. Another thing that makes us really unique, and I would, if you're interested in doing any kind of scientific research, I think this is a big selling point for us. We guarantee research, at least 1 semester of undergrad research, to all of our majors. With the understanding that some students may do more, say for example if you're interested in doing an honors thesis, but we had so many prospective students come to us and tell us, well, how can I do a research experience? And it's going to be really hard for me to do research with a professor if I'm accepted to a university where they may have 1,000 students in a given major, say in a computer science major. How am I going to get connected to one of the comparatively few faculty that are doing research in an area that I want and actually stand out? We have worked with our faculty to make a pledge to all of our students that everyone is able to do research for credit. So this is something we're really proud of. We're also proud that our students have self-organized themselves, and they've formed a student organization for students at CMU, not just in the School of Computer Science, but across colleges for any undergrad student who's interested in computational biology. This is a relatively new development, at least at the time of my recording this, and we really look forward to doing a lot of cool things with them, whether it's student events or bringing in external speakers or having kind of hackathons as part of this student society. We also love connecting companies to our students. So we acknowledge that what companies do in this area can be spread out across the US. It can be a small startup that's funded with a bunch of venture capital funding, or it could be part of a huge $50 billion valuation pharmaceutical company, all right? And so there's a big breadth there in terms of the organizations and the companies that are doing work in the life sciences that's heavily computational. So we've made huge efforts to, as a department, reach out to companies and get them connected into what we do at CMU, coming in and having panels for our students, for example. So this was a panel of representatives from Regeneron who talk to our students about the kind of cool scientific work that they do on a day-to-day basis. These are the places that over the past year and a half or so that we've had on campus or talking remotely to our students about the scientific work that they do. Some of these places you might not have heard of, some of them you might not have, in many cases because we have a lot of new companies. It's a hot area of research where we see companies funded every day. We also love connecting alums to our students. Even though this is being recorded in the midst of a pandemic, we have next week an alumni session where our alums are going to remotely connect to our students and talk about what scientific work are they doing in their day-to-day lives. We try to get a good balance of this. That's actually oversubscribed, so we try to do this at least once per semester. And it's great for students to see at all levels, whether it's an undergrad, a master's, or a PhD student, to see the types of things that people are out there doing in industry or in research organizations, and especially because it's growing so fast. So there's these companies that alums wind up at that have just started and are really taking off. We felt so strongly about this that we wound up building an entire careers website to house information on these organizations in computational biology because no one had done it. And so if you check out careers.cbd.cmu.edu and you don't trust me that there are a lot of different places that do computational work, well, go there and look at genomics in the Bay Area and see what comes up. Look at pharmaceuticals across the US or in New York City, for example, and see all the organizations that come up. You can see there information about educational programs, whether it's a summer research experience for undergraduates or whether it's PhD programs in computational biology, and you'll find about 250 or more, because the resource is growing, companies that do work in this area. We probably wouldn't have launched this entire website had we realized at the start just how many places out there have really cool, compelling, well-paid positions for computational biologists. After all, I always tell students, 2 of Alphabet's companies are comp bio. Yes, Alphabet is famous for Google, but they have grown and broadened what it is that they do. And 2 of those sub-companies of Alphabet are directly connected to computational biology. Verily does healthcare and disease prevention research. Calico is interested in another offshoot of life sciences, which is research into human longevity. This is a moonshot, just as the sequencing all the letters in a human genome, being able to read 3 billion letters in this submicroscopic text that corresponds to what it is to be human. That was a moonshot. That was a dream at one point in time. Well, we have another dream now, which is how is it that we can figure out what is longevity really? Are there ways that we can slow it or reverse it? And imagine how this could shape research in the 21st century or beyond in terms of figuring out the key to human longevity. And I always tell students too, Google built a search engine, and look at how valuable that was to be able to quickly type in a query and get a piece of information back. But how valuable would it be to have a technology that could increase human health by, on average, 10 years? How much would, if you're a student, how much would your parents have paid for such a thing when you were born in order to ensure that you had a longer, healthier life? All right, so there's a lot of interesting parts of biology, and everywhere you look in biology, there's computation. But I always tell students, you don't have to take my word for it. I've got 2 very prominent people here and grabbed quotes from them. Francis Collins is the NIH director and was integral in sequencing the first human genome. And they asked him, well, if you were starting out in college today, what would you do? And Francis said, if I was a senior in college, I wouldn't go the route of being a lab biologist, which is the route that he went. He would just start with computation. He would start his training as a computational biologist and go that way. And they asked Bill Gates, the Microsoft co-founder, what would you do? And Bill said, well, energy is one thing I'd be interested in. AI is another thing. So there's a pointer at our wonderful AI major. And the third thing is biosciences, promising fields where there is a lot of computation involved and you can make a huge impact. We're in many senses where I feel like computer science was at a much earlier age, where there were these exciting problems and people didn't even realize all the exciting problems that were there. So that's my talk, and wherever you are, and I hope that this has been a little informative if you've managed to make it to the end. And I'm very easy to find, so I would encourage you, feel free if you have any questions, don't hesitate to reach out to me. I'd be happy to start a conversation with you. And otherwise, I wish you the best of luck in whatever you do.